Assalamu alaikum everyone. I am delighted to welcome on the behalf of Biotechnology.org. Today, our honorable guest is Professor Dr. Mushtaq Hussain, former principal, Dow College of Biotechnology, Dow University of Tech Science. He is a respected academic researcher and mentor who has made the valuable space contribution to the fields of biotechnology and genetics. Through his research and dedication, teaching, he continues to inspire students and young scientists to pursue innovation and scientific excellence. In today's conversation, we will explore his academic journey, running research, and the role of fly lab in genetics studies, vaccine design, and the impact of artificial intelligence in biotechnology, and his advice for aspiring researchers. Sir, thank you for joining with us.
Operator.
What about you think? What do you think about Biotechnology.org after joining how fail with Biotechnology.org? After joining with Biotechnology.org, how you feel?
Oh well, it's a very exciting uh, proposition as well, an initiative which has been taken by some of the individuals at the Bangladesh, the Brahmadi country of the Pakistan, and I wish them all the success, especially the kind of the forum they are providing to the scientists, the budding one and the mature one as well. So may it be an inspiring beacon for those who wants to be the scientist or took scientific research seriously in their career path.
It's kind of started COVID, which they all the students are too addicted for social media. This time, Dr. Mushio sir, think about this. If anything do, there always was social media. So there was the research case, a very effective case, like any scientist interview, they read any scientist research work, then they can learn anything. They are addicted for mobile, no issue. They're addicted, but they say the any effective items. This time we started, and after that we see everyone response, a both response, and then we continuously our interview program posted, and also after that we can continuously offline program arrange. And then nowadays only in Bangladesh we arrange the program, but then perhaps Bosho sir say you can arrange a global program. We not only contact with Bangladeshi scientists, we tend to have
Understand abroad.
abroad. So could you explain the role of the file in genetic research and why it is important for scientific discoveries?
Okay, so fly lab actually generally we refer to the labs which are working on a very small insect which we call Drosophila. And now Drosophila apparently commonly referred to as fruit fly. They are very small in their size, 2 millimeter in size. And apparently they have no ecological importance. Like they are not the pest of any plants, they are not the effective pollinator, but still the fly holds quite a lot of importance in what we know today about the life sciences. And there could be a variety of reasons. Let's put it in this way, what we know about the human biology or the biology in general is mostly because of the experiments which have been conducted on animal models. Okay. Now, animal models are generally being chosen just because of their genetic similarities. So the Drosophila has about 60% of the genetic similarity with that of the humans. 75% of the human disease genes have an orthologs being present in the fruit fly as well. Then there are some other advantageous traits which the Drosophila has, and that could be used that could make it a very classical animal model for the studying of the genetics. One, they have a very fast recreation cycle. They get sexually mature in about 6 hours period of time, and they get mate. And a single pair will produce about 100 to 200 progeny in about 10 days time. So since the genetics is the matter of numbers and how effectively and how fast you are getting the progeny and how many numbers you are getting, so that allow quite many of the central discoveries of the genetics being made of the fruit fly. That is why it is one of the earliest of the organism which genome have been sequenced, and Then about 500 of the human diseases have been successfully modeled on the Drosophila. It doesn't require too much amount of the money and the resources to maintain, unlike mice or any other animal model.
Also not needed to was a place.
Space, indeed. That gives Drosophila, that base Drosophila is a very ideal position to be used as an effective disease model. So far, about 6 of the Nobel Prizes have been awarded to the studies conducted on the fruit fly. So that's another testimony of their significance in the today's biology.
So can you tell me about your educational journey?
Well, I have been born pretty old, I am, and from Generation X, if you like to say. So, very late of the Generation X and very early of the millennials, if you like, or at the interface of both of those generations. So, I born in 1978, to about 48 years now, maybe. Uh, my initial education is from a very underdeveloped area of the Karachi, which is still quite underdeveloped. It's called Lyari. Probably very recently you may have seen a movie, the Indian movie doing that.
The grandmas.
Yes. So that is the area of that entire movie is based on one geographical location, which is the Lyari. It's been infamous for the gang wars and etc. So I've been educated from there. I spent 30 years of my life at the Lyari. I got matriculated from there. There is a school symbol as a stop. From where I then matriculated from, and then I got admission in the DJ Science College, which is one of the prestigious colleges of the Karachi. So I did my intermediate and BSc from there, and then I joined Karachi University as an MSc student, where I did my masters in microbiology, which is currently being referred to as BS. At that point of time, it is actually the equivalent. It is actually equated with the MSc. Then I spent some time in school teaching. And then I realized that this is not the area where I need to pursue myself. It's not too much of the development into it. So then I started working. I joined Center of Excellence in Marine Biology as a research associate. But in 6 months period of time, I got an offer from Dr. Kifan Institute of Biotechnology and Genetic Engineering. So I spent a year over there as a research associate. Then I moved to the ICCBS, which is one of the premier institutes in Pakistan as part of the research. I spent 2 years over there, and then I came back again to the KJ, Dr. Kifan Institute. Then I spent some time in the ECSIR and then at Afan University. And then I got a scholarship from the Dow University in a faculty development program to conduct my PhD in studies. So I completed my PhD from the University of Glasgow in genetics, genomics, and system medicine. Then I spent some time at Lancaster and Cambridge, and then I came back with Dow University. I was joined as a research associate even after doing my PhD for a year, and then I am promoted as an assistant professor in year 2015, and then in year 2031 I am promoted as a full professor in the patient-based professor. I enjoyed at the same time the corpus of principle.
So you had so many opportunities to shoot it in USA or UK, but why you decided to stay in Pakistan?
Well, let's put it in this way. It's part of both things, to be very honest. I have a bond with it. There was a contingency bond. It's contingent to my scholarship earlier for the PhD that I have to come back to the Pakistan and serve for 5 years until doing my PhD. So that first return is because of that, if you like to say. So I've done my PhD and then I came back over here, so I had to serve with our university for 5 years. That has been done. And perhaps you may say that traffic has been born by that. Because once you're done with those 5 years, you got distant or patent dies, number one. Number two, what I personally feel, and maybe I am doing wrong. In Pakistan, I find myself much more freedom in conducting the studies which I want to conduct myself and which I own myself compared to if I be at that level. I'll be working under some supervisor, which so independency would take some amount of the time. And into Pakistan, fortunately, I was able to get it much earlier. So it's all good that I stayed in Pakistan. So the
It is not. We already say most of the scientists not returning their home. You either Bangladesh is a common thing, then you go outside of country, you can hard to return for study purpose.
same applied to the Pakistan in cases. Yes. But to this time, I have heard. Many of my other colleagues have also written.
So could you share some insight into your current research project and their visual impact?
So I actually heading a lab which works on the Drosophila.
Yes,
So as you may have already judged it by now. So there are quite many of the projects which are going on, but two or three of them are very exciting one. That's it. The first one we have isolated and successfully cultivated endosymbine bacteria from Drosophila. It's called Wolbachia. Now, Wolbachia has been effectively used and been proven to be used as a biocontrol agent against insect-borne diseases. So, because what happens that the insect is being infected with the Wolbachia, that it crosses with the uninfected insect, the progeny would be sterile or progeny would simply not be formed. So say for example, if a mosquito, which is going to be basic carrier of the malaria and dengue and chikungunya and many other diseases. So if one of the mosquitoes has been infected and the other mosquito is not being infected in abdominal hours, what's going to happen? That the progeny would not be formed. So that bacteria could act as a biocontrol agent. So we isolated the bacteria from the Drosophila, we cultivated it into the embryonic cell line of the Drosophila in our laboratory, and we sequenced its genome. And the next target would be that to use it as a biocontrol agent. That's number one. The other project which we are running is about the traumatic brain injury.
I see already.
Yeah, okay. So what we are actually exploring to that the kind of the genetic variations that can make individual be susceptible for the good or a bad prognosis of the TBI, traumatic brain injury. So say for example, you have been hit on head, but you survived okay. But if you've been hit by the same intensity, some individuals have been hit by the same intensity, they won't survive, or they got some other debilitation or incapacities, like they got stroke or they got paralyzed, etc.
Also die.
Sometimes also dies. So we thought that that could be because there could be genetic underlying in that differences. Certainly, you can't conduct that study in the humans because of the ethical reason. In the mice, the numbers are not enough to conduct that study. So we choose Drosophila. So what we are conducting, we are actually isogenized 25 different wild type lives from different parts of the Karachi, expecting them to be genetically variable. We subjected them to the traumatic brain injury by a particular device. And we have identified some of the flies are very resistant to the traumatic brain injury. They call it the traumatic brain injury, but it survive okay in about 80%. And some are not surviving okay. So we are sequencing your genome to see the genetic differences between these. And once those genomic differences have been found, then we compare it with the human gene, the counterparts which are present in the human. So those genes can then act as a marker for identifying the best of the individual for the contact sports, kiteboxing or Muay or kickboxing, or even it does a military application as well. Who to send the front line and who to send, who to be busy with that. So these are the two projects which we are running at this moment. Third, we are actually using CRISPR and RNAi, the gene editing tool, the RNA interference, to model different congenital heart diseases, the heart problems in the child, which is due to therapeutic reason within the Drosophila. So these are the three major projects which are really very interested.
So in our country, people think about it is the developed countries provide free vaccine as a trial to the developing countries. So can you explain the HC design further?
Well, that may not be exactly true. There is maybe so. If I'm getting your question correct, that they develop the vaccine and the trial has to be done in the Pakistan or anywhere in the underdeveloped country, and then they will be applying it to their own population or not.
Yes,
So they
yes.
are use it as a guinea pig
Very
in like that.
yes. Very
That
smart.
it's not true. It's not true. By and large, not true. I've never seen any credible witness or evidence in this connection. And case in point is SARS-CoV-2.
So, can you explain the vaccine design process?
Okay, so it depends which kind of the vaccine which we develop. The most conventional way is the live vaccine. That is our College of Biotechnology, as you might also be aware of, that we have developed the vaccine, live vaccines, which is of the virus of poultry, and DC flyers. So it's the most easiest and the most conventional way of developing the vaccine design. Say, developing the vaccine. So you take a virus or a pathogenic agent, you treat them with certain chemicals or passage them to the extent so they lose their disease producing ability, but they still hold the immunogenic potential. So the virus or the bacteria once lose their disease causing ability, they are still alive, but they have an immunogenic potential. So you introduce it into the individual, what happens to the virus or the bacteria may not be able to create the disease. But because of their immunogenic potential, so antibodies will be formed and the memory cell will be there, and this will activate the vaccine. This is the most conventional way of developing a vaccine. Over the period of time, there are other forms of the vaccine that will develop.
What is dry process to process?
Yes, that's one of that is the mRNA vaccine, the most modern of the form. And it has vector's own advantages, like because of the mRNA vaccine, and you can cheat in the pathogen is changing their immunogenic potential or their antigenic traits. You can easily fix it within the mRNA vaccine. And initial process is entirely dry. You can use bioinformatics or deeper to the AI tools to develop and design an mRNA vaccine. I'm not sure whether this is true or not, but I've seen a few days ago a read where an individual who has no bigger degrees in the biotechnology or biological sciences has actually used AI to develop an mRNA that can treat his dog cancer.
Doctors.
Yes, his dog got a cancer. So he took some samples and he analyzed them through the laboratory. He got the results. He provided that results to the some AI server. I'm not sure whether how true it is, but I've seen that read being scanned through my uh, eyes. And similarly, there is one other individual I've heard of. One other child which I've heard of. He designed an experiment using AI to actually see whether the butterfly holds caterpillar memories. It's exciting to know the potential of the AI in that. So, so the mRNA vaccine could be developed using tools of bioinformatics, in which what happens, you develop a single mRNA that includes the part of the epitope switch of any pathogen. And then the mRNA, once it's designed, then it goes to the nucleic acid synthesizer, where this entirely mRNA fragment has been actually developed. Then it has been encapsulated into the nanoparticles or nanovessels, and then introduced into the individuals. So once it goes into the individual, that nanovessels or nanoparticles obliterate themselves to release the mRNA, which penetrated into the B cells, and from where the B cells, which are supposed to capture the antigen, will actually start producing the antigen without the whole virus. So it means now your immune cells can recognize the
Vivo test.
Yes, yes. When you can conduct similarly the in vivo tests which have been conducted for the SARS-CoV-2 mRNA vaccine and the other mRNA vaccine effectively. And because of their pharmacy results, it has been first employed in the UK and USA, and later it has been shipped to the Pakistan or any other part which has suffering from the COVID-19. Yes. So trial has already been done there.
After in vivo tests, always preclinical trial and clinical trial by conducting with the doctor and other expertise. After that, it's government
Approved.
approved.
Yes.
So it's not a
It's not. It just can't be happening like that. You may make a conspiracy theory out of it, but it does not work.
general people always think about this, and after that they make awareness against vaccine. But we always say vaccine always a blissful thing.
Yes.
After COVID, after take COVID vaccine, or maybe Pfizer or another, some people face the allergic reaction, and then they spread this type of report.
So it's always be for any drug or for any vaccine. It's always be a trade-off. So there would be few side effects.
Maybe
of
the
course.
immune system changes after COVID.
That would be one thing, and because there is an increasing evidence now that those individuals who got the mRNA vaccine have a higher risk of getting heart. Now, hold on a second. So, you need to see this way. What else we have as far as SARS-CoV-2 once that pandemic was there? What else we have? This is the only vaccine which has been available, the effective vaccine. People have taken. So on the one hand, it might increase your chances of getting heart attack by 0.1%. On the other hand, you may have been already dead because you haven't taken the vaccine. So which one? So any drug or any vaccine may or may not have some side effects. So you have to weigh that the advantages are more or the disadvantages are more. And based on that, we make the decision. So by all means, if I have to give, let's say that the pandemic will be there, and you introduce, and the vaccine has been developed by the mRNA or any other means, I would go without even giving a second thought. Doesn't matter.
No,
because
you don't need the second part.
No,
It's a very-
yeah, because the whole history of the human civilization is a witness that the vaccines works. It prevents the disease to happen. It prevents now even hemorrhagic fevers like Ebola or other Congo fever to be spread across the African land. And now it also curtailed HIV to a significant scale. So, there is no question about it.
So was it the major challenge of obstacle that scientists and researchers still face in models? Research student.
Well, I would always say challenges are a bit of an opportunity, if you like to see. So the challenges as far as a researcher over here in the biotechnology is like many other countries of the world, fundings, availability of the college, because the research mostly won't plan without the proper permission platform. Consumables,
Vaccine design very costly.
vaccine design or any other research for that matter, because most of the consumables, particularly the chemicals which you want to have in the research project, are imported, and it takes some amount of the time. It takes quite a good burden over the finances as well. That's number one. Number two is a certainty in the policy. So say for example, I'll take an example of the Pakistan. At one point of time, and I can see the reason why it's so, because at one point of time from the era of 2002 to 2007, this is the time where the Higher Education Commission in the Pakistan had been built. They invested quite a lot under the leadership of Professor Dr. Ataur Rahman. They invested a lot in building the universities in the Pakistan, the research infrastructure. They put hefty salaries to professors. Otherwise, our professors are not getting too much amount of the money before Professor Ataur Rahman. Neither the current the architecture which you see around Pakistan as well, the research infrastructure is concept, it was not there. It was simply not there. It was merely because of Dr. Ataur Rahman. But then we are witnessing not a plateau that you reach to your optimum and then there is a plateau. No, we are gradually, with each passing year, we are observing a bit of a decline in the already established research culture in Pakistan. And you can blame it to multiple different reasons, and some may prioritize one reason being more, and the other are prioritizing other reason are more. But that's what we all are being actually agreeing, that there is a decline in the quality of the research, the amount of the research, or the type of the research we produce in the parks. So, and it's because of the lack of funding. Right? Lack of funding, and the incentives are moving in some other directions. So pupils is more of a number game rather than actually the quality being produced. Yesterday I was last night. Yes, last night I was reading an article over there. Guess what? How many number of articles? Nature Scientific Report. That's one journal I am talking about. That's one journal I am talking about, and we actually produced last year. How many? Guess. You guess or not?
How many?
So it's a very good scientific journal, which is called as Nature Scientific Reports. Okay. How many number of research articles being published in the Nature Scientific Report last year? Surprisingly, it is about 50,000.
50,000?
So that's one journal I'm talking about.
Are it one journal?
One journal.
50,000?
50,000 scientific articles have been published. Nature Scientific Reports. That's one journal I'm talking about. So from there you can get an idea there'll be a need. Okay. And if everybody is doing the same thing, then actually nobody is doing anything. So, and similarly, there were states like that for the one, and there were states like that for. There is a journal which is called as ELAB. In the same article, it comes through me. Yeah, because the memory works for fine, because I just read it last night. So ELAB actually the elderly impact factor, and now the Web of Science, which is the authority of the journals, actually classified them as a paper. So these are several challenges in terms of publication, in terms of the grants, in terms of the quality of the research, but these are also at the same time the opportunities as well. So we have the challenge being there. Let's work hard, or work in a way, or navigate around it to circumvent or to rectify that challenge, or to address this challenge per se.
It's like, how has artificial intelligence transformed biology?
Well, quite a lot. Quite frankly speaking, and we're just seeing the beginning of it.
Biotechnology is preparing this is on action.
But in part, yes. So, say for example, the role of the bioinformatics has already been established in the vaccine design and drug design.
Okay.
So the both are the areas comes under the compass of the biotechnology. Since bioinformatics, that is the computational biology, actually has an instrumental role in developing the designing of vaccine or designing drugs. And with the advent of the AI being there, that has been increased significantly. So one of the two major flags of the biotechnology, that is drug and the vaccine design, is being actually influenced by a lot by the AI. Similarly, if you go further, let's say, now you can put any kind of the algorithm or the data, or you can train your machine with any sort of data. Say for example, you use 50 different combination of the nutrition of fertilizers or fertilizing elements, and you treat it with the, let's put it in this way, different soils for the plantation, and you found certain results which are being ambiguous. Now you can put all of the data to the AI web servers, and then they can spew out the optimum combination which you've been tested in such a little time, which you may have taken about 10 years to conduct that study. So yes, it has been actually changed that landscape of the biotechnology quite a lot.
So, viewers,
Yes, indeed.
Last day,
It diversify in multiple different dimensions.
last day I can read the Amazon work workshop. Eliza, Eliza. This time I see also needed a additive US compared to the
Titles
and here
the amount of range producers.
and your interests. Then art instead build a successful career in biotechnology. Because most of the students read about this now. I studied in biotechnology after the part I too. Which sector I go, industry or pharmaceutical sector or teaching profession, which thing I can choose, which is a very confusing. I also think about it example that what I do is.
So as we move along, at one point of time, let's say back in year 1950s and 60s, you may say if you get a matriculation done, you may find a very suitable job. Okay, that thing has been gone to has been advanced to intermediate when we go into the 70s and 80s, and then it has been advanced to the bachelor level in the 80s and 90s, and in the 90s at the master in the millennium after 2000, at least masters needs to be there. Yeah, post edition has to be there. So one thing is that the future of the biotechnology or establishing a career that making the biotechnology.
I belong. This is an issue.
Yeah. So it all depends whether how advanced or how far you are getting to go. Number one. And number two, it also depends on that what is your interest. So say for example, if you want to go into the industry, yes, you have quite a lot of opportunity in the industries as well. Say for example, you may have already witnessed it, that we have run industrially funded project at the Biotechnology, at your own college. There is a College of Biotechnology, and we hired 3 individuals as a research associate. And now, at this moment of time, they have further extended this project to a completely different scale. And now they are paying for 5 individuals to be get trained over here. And then after September, they have already a job being placed at the industry. So, yeah, 2 of the individuals from here will be placed over there as a biotechnologist to conduct the study to actually expand that the project which we have successfully conducted over here to the industry over here. So that is, it all depends how consistent you are, how willing you are to be focused, despite you may not get the earlier success, but if you remain consistent enough, then there is all likelihood, and if you have been dedicated, you can find your placement there. Doesn't matter which field of the biotechnology you pick. You can pick industry, you can pick teaching and research as well. You can go to an industry which is not of the pharmaceutical nature, let's say the sugarcane industry or in the sugar industry or in the textile industry. There's a whole lot of room for biotechnology to be placed. One of the recent emerging track in the world is actually the environmental. Okay. So we have Dr. Faiza over here who works on LV, bioremediation LV, with reference to the bioremediation. So that's another area where the biotechnologies could be very instrumental in the next decade or so.
So many prints in it, sort of.
Will emerge and the biotechnology will be easy compensate. Absolutely.
So good about you. So one last question is, always that's you will to the new generation.
So it's more of a derivative from the different kind of the thought, rather than my original thought, or partly you may say it my original thought. I believe do whatever you want to do. It doesn't matter. Don't worry about the scope. That the thing which looks very lucrative at this moment of time, as you are seeing, it may not be as lucrative in about 5 years' time. Much has been, and especially emergence of the AI. You may by the same token, let's say at this moment of time, people are saying, "Oh, everybody is going to the AI. Oh, let's learn AI because it has an scope. It has a scope." Well, it may have an scope, and it does have an scope. But how about thinking in this way? AI would replace the AI creator.
Sunday, so they control our mind also.
Yeah. We did AI, we just designed AI to develop another AI program to develop another AI program. So it's all layer by layer here. That's one. But let's not rush towards it just because you find it, it seems like that there's a job security there. I personally believe you have to do what you want to do. You've got one life, about
I
60.
is not wanted to do anything in my field. It's not possible I can do anything.
You would not excel in it. You would be another mediocre in that particular field, and you would not shine bright. So you have about 60-65 years of your life. I think so. Do what makes you happy. Except for that, if your entire orientation is money based, then yes, do something which could generate a lot of money. If you want to have a fun and a hobby and everything in combination, and which excites you as well, then yes, choose whatever you love. Let's say if I'm excited about science and running experiments, I have to do this thing. It may not generate that much amount of the that's for sure. As if you're doing a business or something around your banks. Absolutely. But it can give you other kind of the satisfaction which if you will be doing a business, you won't be able to get it every year. And you will get boredom in about 45 years or 50 years of time, or by 55 years you will be thinking, gosh, perhaps I had done that. And then I think about this God and doing my podcast. Yeah. So that is what my point is. At least I die very satisfied. I get what I want to do.
Just a please, later.
Yes.
Now our question and answer session for our audience. Our audience throw question to the professor, and professor answered the question. Well, the information you gave actually is so interesting to me and can despair for more degrees. I'd like to ask, what should I do to get admitted to top rank university for my higher studies?
What should you do?
Yeah.
Well, it depends on like different university prioritize different things, and they want to see different thing in their prospective students. Let's put it in this way. So say for example, Cambridge may be looking at the innovative side, and the University of Glasgow may be looking at the sports side. So first you target which university you want to go for, and look at their research programs, where they have been more focused on. So say for example, if you get your education, some certification, or get your skill sets being developed in that particular research themes which the university prioritize, okay, you got a much brilliant chance of getting admitted into that university. So say for example, there are 100 disciplines of the research. Entomology could be the one, oncology could be the one, vaccine development in immunology could be the one. Let's say epidemiology could be a one. Every university you are targeting is more interested in the epidemiology and find they have a very better or very strong epidemiology program, since you are from the nutritional sciences that needs it. So in that case, you should be working in the area of the epidemiology, and you'll be applying at the area of the epidemiology because most of the university resources are going towards the epidemiology in that particular university. Okay. So let's say if you attend during your tenure of your BS or graduation some conferences of the epidemiology, you may get met up with some individuals who can make you research, and if you with the epidemiology, this can join them, participate in their research. That's how you can develop a skill set. And if you be lucky enough and your data is good enough, you may get a publication during your graduation, within your graduation time.
Did my
Yes, absolutely.
Okay.
And that will seriously extended or increase your chances of getting admission in any university of your life. The first target which university you want to go, which 2 or 3 universities you want to actually tap on, and look at the strongest of their teams, because that's where most of their sources, financial and administrative sources have been amounted to. And if you excel in that particular area, those universities are more welcoming to you compared to
Others. Yeah. Thank you so much, son. Junior.
some of the fishes, which is about 300 to 400 million years ago, being buried between the slabs of the rocks. This is a trilobite, one of the most common possum you will find in the Cambrian or post-Cambrian
From
era.
very courageous. From very courageous.
These are from the United States. This is from also from the United States. This I bought from US, UK,
Okay.
but my best guess is that it is also from United States or China. And these are the bones of cephalopod, like you have seen sepia and loligo.
Well,
These are cephalopods.
like calamari.
Yes, okay.
So calamari or some I don't know squid.
Squid.
Like this.
Yeah.
Calamari or squid.
So they have a central bone, which generally in the living animals, literally, but
This is a recent
recent, the extinct.
Okay.
But their extinct species have a bones which have been actually fixed.
And this is a fossil. How many years have passed?
Roughly around 400 to 500
400 to 500.
million years.
Million years.
Oh my god.
Wow.
So I have it.
This is Nautilus. This is a mollusk. Again,
a mollusca.
the girl from the these are also mollusks as well, but these are from the cephalopods. These are from the gastropods.
That's right.
Okay.
I also tried to record because the previous time I did Dr. Major once. This time I see voice is not clear. So
Did you have mites?
no.
Oh, we do have mites if you want.
Okay.
My PhD student.
Who?
So for the work, we were working on the cerebellum.
Okay.
So we dissected that monkey.
Okay. Well. And then collected the bones.
Well, we have that bones I have collected for myself.
Oh.
Their work is majorly on a cerebellum. So it's a part of the brain.
Okay.
So if it is,
Neuroscience.
yeah, something like that. These are the sharks, these are the frog, and this is of cat cerebellum, and this is of the snake cerebellum. Oh, sorry, monkey cerebellum.
Monkey cerebellum.
please.
Yeah.
So the bone which you may be seeing over there, the first one, the white color one,
Yes.
that's the male vertebrae.
Oh.
It's monkey. Now these are the bones of humans.
It's
Yeah, you may pick it.
A skull.
So yes, skull. This is a femur.
femur.
This bone,
Yes.
Femur.
and this is a pubic. This is again a part of the skull, so you can see the human, and that's where the
Another.
recovery comes. We just broken a bit,
Okay.
but
Yeah.
this and the monkey bone are present here in this.
So
Okay.
it is a total human body.
Yeah, human body.
The human is going to be
Monkey too.
Slightly bigger than yours.
slightly bigger.
So if you may, so this is supposed to be present like this.
Okay.
So is it bigger? Like
Yeah,
it perfectly suits her, no?
she is about, let's say 5'3",
5'3".
5'3".
Yes.
So that particular person may be 5'6",
5'6".
Oh.
5'6", 5'7".
Okay.
And it is male. 40 centimeters, like thick.
Not really, because the thing is that we have is unregarded.
Apparently, turtle always
Of
always
the
has
same,
finger.
yeah. So
What is
we
it?
know because they are the same human. So we know this is actually the male one.
Okay. Always a woman has pretty bit of bigger size than the
Yeah.
little that difference.
Coincidence, but it doesn't really.
None of us not gonna do anything.
Hello.
You are not coming there.
That's terrifying, but I love. Hello. What's his name in his series?
It's Alexa.
It's Alexa. Okay. Won't it bite?
At times, but generally just a small scratches, nothing more than that.
It has something strong here, isn't it?
Vertebrae. Vertebrae.
Oh, okay.
That's vertebrae and its ribs.
Your tongue is so nice. Thank you, sir. I can't more. Thank you.
This is fly lab now is to anesthetize the flies for their researching Prof. They separated the flies after anesthetize. The anesthetized flies by using ice cube, and then it's totally anesthetized and separate the male and female. Well, this is incubator. For which purpose this use? Can you tell me?
Basically, it maintains the temperature and also the circadian rhythm during day and night. Alright.
And what is this?
This is the microgravity. This is for the microgravity, the inducer, basically. This is a climbing stone, 2D climbing stone. Okay, done.
Okay, this is houses society of flies. The Drosophila melanogaster means they live in a controlled environment with controlled temperature and humidity conditions. And what is this? This is microgravity equipment. This basically simulates weightlessness conditions on Earth, which he experienced in outer space. And nowadays, you started your research about Drosophila. Yes, he worked on model organism, Drosophila. Yes, this classic model organism, Drosophila melanogaster. And this lab is conducted by Professor Mushtaq, and you all work under it. Yes, we work under it. He's the supervisor and boss. And I'm fly lab curator. Fly lab curator. She is Dr. Aisha Abdullah. She's the fly lab curator. So today I visit all fly labs and another labs of Biotechnology department, and also talk with Professor Mushtaq. So I would glad to have to see this lab and observe the lab's tools and other things, and it's a very curious and also enjoyable moment for me. Thank you, ma'am. Thank you so much. Thank you.